students, in the previous lecture I introduced you to different proteins, how they behave, and then we talked about cells growth. So, how can we humans know where the cells are in their replication process, how fast they are growing? Today I would like to revisit the model for cell replication, because I think it will help gain some extra clarity about what is happening in the when the cells are growing. So, as a recap, in the previous lecture we talked about how cell undergo binary fission. So, this is a typical cell and it will elongate, create a septum. So, septum is that place from which it starts tearing into two cells, and then lo and behold, within one generation time we will have two microbial cells, two daughter cells where there was only one parent cell. So, we noticed that to begin with we had 2 to power 0 cells and then 2 to power 1 after one generation time, and then after n generation times we will have 2 to power n cells. Now, in order to model this cell growth, and I mentioned it previously, one way we can go about is take a general time when n generations of time has elapsed, and find out what the typical microbial rate growth would be, which is depicted by del n by del t. Here n is the number of microbial cells, t is the time, and remember we started with n naught or 1 cell. And here we can say that n generations have passed. What it means is that the time in this moment is actually n into g, where g is the time it takes for one microbial cell to replicate under ideal conditions. So, del n by del t, at this particular time when t is equal to ng, the number of microbial cells that we should have ideally will be 2 to power n, and in within next generation time after time g, in an interval of time g, we will end up having 2n plus 1, and thus del n by del t will be equal to 2n plus 1 minus 2n by g. Now, obviously this can be very easily solved by taking 2 to power n common, and then we have 2 to power n, 2 minus 1 upon g, and thus we are left with 2 to power n upon g. Now, notice here 2 to power n, which is the numerator of this right hand side of this model, is actually the number of microbial cells that we ideally have at this particular time already, and thus we can also write this as n by g, right. So, to simplify, now our model looks like del n by del t at any given time is equal to n by g, which is number of microbial cells upon generation time, correct. Now, 1 upon g can be 1 upon g, we can give it a different constant. I mean, we can write it as a different with different notation and refer to as Rn. Now, note here g is the generation time, thus g is the time it takes for cell to replicate. Its reciprocal would give an idea of how fast cells are replicating. An example is, if it takes you 3 hours to do a homework and it takes me to do the same homework, it takes me 5 hours. So, I take 5 hours to do a homework, you take 3 hours to do that same homework. If we do reciprocal of our time that it takes for us to do the homework, we will get an idea of who is faster in doing the homework. For me it would be 1 upon 5 and for you it would be 1 upon 3, and very easily we will know whose rate is faster. Your rate would be much faster than mine, and thus R is also referred to as rate of replication. And as a teaser, I asked you to tell me what is one major obvious limitation of this model. So, today I want to visit briefly the major limitation of this particular model. So, right now we have the growth model for binary fission of microbial cell as dn by dt is equal to Rn. So, let us integrate this to get our equation for microbial growth, and in order to integrate this we need to separate our differentials first. So, we can bring dt here and we are left with dn by n is equal to R dt. Now, integrating from time 0 to t and n naught to n microbial cells. Now remember, at time 0 we started with n naught microbial cells, at time t we have n microbial cells. So obviously this is natural log, and here we have RT. And thus we know now that number of microbial cell at any time will be equal to initial cells that we started with into ERT. So this is an exponential growth. Now with any exponential model, whether it's exponential growth or exponential decay, as is case with radioactive materials, we have a concept of half-life, or in this case, because it's a growth model, we'll have a concept of doubling time. So how much time does it take for microbes to double? And it should be very easy for you, because for doubling time you would know that n by n naught will be equal to 2, will be equal to ERT, and if you know the rate you can easily calculate the time. Or if you know the doubling time, because you have a mechanism for counting microbes and we talked about that mechanism, then you can actually find out the rate. Now if you plot this model, it looks like this. You start with n naught, and then you have an exponential growth. So if as you increase time, you will notice that microbial cells will grow very large in number very soon. In fact, there is a particular strain of E. coli which is predicted that if allowed to follow exponential model with just few microbes, in 48 hours it will have more mass than the entire earth together, which is obviously improbable and impossible, completely impossible. So there is a major limitation with this model, which is that it expects that as microbial populations increase, nothing will change in the environment. But we do know that as number of microbes and their density increases in our media or in our environment, the resources reduce, and when the resources reduce, another factor comes into picture. So now they are not just replicating, but they are also dying because of competition and lack of resources. And thus in our model, we have a need now to add another term that accounts for this decay. So let's do that. So our original model looked like this: dn by dt is equal to Rn, right? And this is our growth term. And as I mentioned just now, we need to add a decay term. So in order to add a decay term, let's think about it. What will our decay depend upon? Our decay will, I know it will depend on two things, but let's explore what it will depend upon. Let's say we have two microbial communities, and at any given time one has n1 number of microbes and the other have n2 number of microbes at any given time t, and they have exactly the same resources, but you know this, that n1 is much larger than n2. Now tell me, which one will have more decay going on in their microbial community? If they have exactly the same number of resources but n1 is larger, intuitively we know that n1 will also face a higher number of decay in their microbial population, and this is actually what we have observed in lab. So we do know that this decay term has to be proportional to number of microbes present in the cell. Now let's look at another instance when both microbial communities, there are two microbial communities having exactly same resources. One has n1 number of cells and the other also has exactly same number of cells. However, there is a difference. n1 is growing much faster than n2. So n1 is consuming resources at a faster rate than n2, right? In our current and more common day life, we can draw an analogy that if there are two cities, both have exactly the same population and exactly the same resources, but one of them is consuming the resources that they are endowed with much faster than the other. So tell me, which city is headed towards destruction faster? And we know that if n1 microbial community is consuming resources faster, then its decay will also be faster, and thus we know that decay should also be proportional to dn by dt. Now because it is decay, it should not have a positive sign, because positive will add to population. We want to reduce the population, and hence we will have a minus sign here. And in order to remove the proportionality here, we will add a proportionality constant. Let us make it k. So minus kn dn by dt. Now this dn by dt is the rate at which it is growing, not the rate at which the population is having its growth, Remember, in biology, growth includes both replication rate and decay rate. So technically this is replication rate. So dN by dt being the replication rate can be replaced by this first term Rn. So now we have our decay term minus Krn square. And this is our new model. And many of you might be aware that this model is very popularly called as logistic model. It serves really well for simple populations that actually undergo either binary fission or have other similar comparable simple process of replication. But whenever we start talking about insects that have a very complicated lifestyle, for example mosquitoes, some part they need to spend in environment, some part they have to do and deal with other hosts, not hosts, sorry, they have to deal with other mammals and animals. And then we have some other insects that actually require to live within a host, such as many worms that live in our intestines at times. So they have very complicated life cycles, and we notice that with them the logistic model doesn't hold true. But for simple growth such as microbial growth, logistic model is pretty nice. Now let's try to understand what this logistic model is. So here we have dN by dt is equal to Rn minus Rkn square. We can write it as Rn 1 minus Kn. Now this is a differential equation, an ordinary differential equation, and in its first order ordinary differential equation, in order to interpret these, one of the easiest way is to make dN by dt equal to 0 and then use a geometric approach. We do not need to study that, but many of us perhaps are engineers, and because and some maybe we are not, but in any case I think it would be a nice idea to revisit geometric approach of understanding these models, because we do not need to integrate it. And it's a good idea to understand how the model is actually related to what we are observing in life. So we have dN by dt, we make it equal to 0. This equation will be equal to 0 only when n is equal to 0. So if n is equal to 0, this term becomes 0, or n is equal to 1 upon k when this term becomes 0. Alrighty. So if we make a diagram with y-axis being represented by rate at which population is growing and x-axis at number of microbes that are present at any given instance, we know that two important points will be 0 and 1 upon k, because at these points dN by dt will be equal to 0. And a very quick understanding will tell us that r is always positive, right, because g is always positive, time is always positive. So 1 upon g equal to r is also positive and k is also positive, and thus this will always be greater than 0, this can never be negative. As long as n is between 0 and 1 upon k, this will be positive too. So we know that between 0 and 1 upon k, this function however it looks like is positive. And we also know that once n goes above 1 upon k, it will go negative. Obviously population cannot be below 0, so we can actually rule out any analyses for n less than 0. So here we know that the rate of population growth up to some instance between 0 and 1 upon k will be in a positive direction. So the slope here would be plus, and thus we will see a U-shaped growth. And once it crosses this particular point, we do not know what that point is yet, but once it crosses this point, then the dN by dt will start reducing. So we will see a growth, yes, because it is still positive, we will still see a growth, but the growth will slow down and it will look like this. And then once population reaches 1 upon k, it will stop moving. So once the population has reached 1 upon k, it will stop moving, and graphically we can now interpret this data as so let's plot here. This is time, this is n, and this is 1 upon k, and we are starting with n naught, which is much smaller than 1 upon k. Alrighty. So up to a certain point, we do not know what that point is, but up to we do not need to know right now. Up to a certain point, the population growth will be like an like a U shaped curve, and as it starts approaching 1 upon k, this is how we will observe it. So without solving the equation, we already know how microbes will grow for a given n naught. Now because the population here stops at 1 upon k, we know that population will not go above 1 upon k. But what if we start with microbial cells that are more than 1 upon k? Let us say our n naught is much bigger than 1 upon k. So here when n naught or our beginning n naught, let us say here, is much bigger than 1 upon k, which is this particular point, we know that the growth rate would be negative. So this is n naught. Let me make this more clear. Okay, here this is n axis and this is n naught. So at n naught we know that dn by dt is negative, and hence instead of growth we will have a decay, and we do not know how it would be, but the decay probably would be like a U-shaped curve, left side of a U-shaped curve, and thus we know we will decay until we reach 1 upon k, and once we reach 1 upon k the population will stop reducing and it will not grow either, and thus we notice that 1 upon k is quite a stable place for microbial population to be. It neither wants to be greater than 1 upon k nor less than 1 upon k. So if n naught is greater than 1 upon k, it will stabilize there. So we notice that as long as we do not have no microbial cell, because if we do not have any microbial cell to begin with, we cannot create microbial cells, right? No spontaneous generation here. As long as we have any number of microbial cells, sooner or later we will all end up at 1 upon k population, and thus 1 upon k is referred to as the carrying capacity of the ecosystem. And we can give it a new notation, a capital K. So K is the carrying capacity. So in our model we write it as so this is the logistic model of microbial growth. So even in logistic model, my dear students, we note that at least if n naught is quite smaller than the carrying capacity, so let us say this is carrying capacity K and our n naught is much smaller, so we can write it as n naught is much smaller than carrying capacity. It does not have to be very small, but it is small enough, then at least for some quite good duration we will see an exponential growth or something that looks like an exponential growth, and thus our first model, which was an exponential model, will be quite correct. So for first some time, in this case we will see our first model dn by dt is equal to Rn holding very true, and thus the population would look like n is equal to n naught e to power RT, which is an exponential growth. After it crosses a certain threshold, and for this course we do not need to know, but if you are interested I highly encourage you to go and find out what the threshold would be. After that threshold it will start looking like an inverted U, and then it will plateau at k, and this is where we call it the logistic portion of the curve, where the decay portion will become more dominant, and this will be definitely a logistic growth. So to model this we need a logistic model. To model this we can still use our exponential model, and this is very important because when we grow microbes in lab, we usually do not have very high population. We usually prepare a very clean media that is devoid of any contamination, especially microbial contamination. We put plenty of resources in it, lot of food, just the right environment. If it is an aerobic microbe, we put oxygen in it, like it is well aerated. If it is a microorganism that likes to live in less oxygen spaces, we reduce the oxygen, just perfect environment for the microbe. So resources are not a limitation. The initial inocula, even though it has healthy microbes in sufficient population to kickstart the growth, but it is not a lot compared to the carrying capacity, and thus in laboratory for quite some time we actually see an exponential growth. And so if you come to my lab here and try to model the observations, you will notice that you will have a growth like this. So n by t, a very exponential growth, right? Now if I ask you, okay, but when you do the experiment you won't get a line, because you cannot take data for how many microbes are present at every given instance. In fact, we cannot gather data for every given instance to begin with in most microbial assays, but what we can do is we can get data points. So it would look like this, for starting you have a data point here, you started with n naught microbes, and then you know that okay, after 10 minutes I had this many microbes, and then after 20 total minutes I had these many microbes, and then after half an hour these many, and so on and so forth. So notice here, some of your data points will fall exactly on the ideal model, some of them will fall up and down, which is expected, totally expected, and it could be because of the error in our reading, it could be just natural variation of microbial growth, or n number of factors we really can't understand completely, but more or less it will fall in an exponential curve. Now if I ask you, okay my dear students, at these data point you know what the population was, tell me what's the growth rate, what is the value of R, and obviously at this stage assume that it is exponential growth, so you will completely denounce the decay part and you will only focus on Rn. Now how can you use this data to find out value of R? Now here is the thing, we know the model n is equal to n naught e RT, e to the power RT, and you have so many data points, and I have had students who just plug in the values of any two data point that they think look really good and they are sure of, and then use that to calculate R. This is not a right way to go, because your R might differ depending on which data points you choose. One very easy and very convenient method of finding out R is to actually plot the same graph on a semi-logarithmic scale. Semi-logarithmic graph. So fancy way of saying that in this particular graph your x-axis follows the decimal system. So we have 0, 10, 20, 30 and so on, and your y-axis has a log scale. Now log scale is very hard to draw by hand, at least for me, so I will not draw it, but I'll show you a slide and I can tell you what looks like an exponential curve will very soon look like a straight line. And why does that happen? Okay, let's take a look here. So here we have our model n is equal to n naught by ERT. Now time is still in the same old decimal system, but n is now represented as in logarithmic scale. So now we have log n is equal to log n naught plus RT. So we will basically just take log of this model, and obviously this is a straight line, this is the intercept. So this is log n naught. This slope of this line should be R, because if we compare it to y is equal to c plus mx model of line, then this is y, log n naught is constant and RT is mx, where t is x, r is m is the slope of line. So if we plot it in a semi-logarithmic graph, we can get a straight line, and very quickly we can find the slope of this line, and this is very easy. You can do it on Excel, and you can also do it manually by giving a best fit line. Usually our vision is very good at linear analysis, seeing straight line and simple patterns. So this is much easier to do. So here we have a population growth and some data that is observed in lab, and you can notice that even though we notice quite a straight line, by the way this is a semi-logarithmic graph. So y-axis is log of number of cells per ml and x-axis is time, and after one data point of minimal growth, we notice that we have a linear increase in population, and even though it is not a perfect line, you can very easily fit the best line and find the R. So this is an example of what I was trying to draw and show you, that on left side of this slide we have growth which is exponential. We plot the same data, remember same data, not new data, on a semi-logarithmic scale, and we have a line, and we can very easily find out the rate of microbial growth. And this is something I already discussed in the previous lecture, that it grows in this way, n is equal to n naught 2 to power n, where n is number of generations. And now this is very important. We notice that in microbes initially they have a small lag phase, and if you go back to the first diagram that I showed you today, there is a small lag. The first two data points have minimal growth. So initially it is referred to as lag phase, where microbes are very sluggish. They are still trying to settle in, warm up to the media and get used to the environment, and then they follow an exponential growth, which is modeled by the exponential model that we just showed you, and followed by a logistic curve, and then it plateaus and it stays so in its carrying capacity for quite some time before it starts dying. And how do we know this population growth? How do we observe it easily in lab? We measure turbidity. So this green line actually depicts turbidity, and that's what we measure. And we assume that number of cells that are actually alive and kicking can be represented by the red line. So red line is actually what you're noticing. Because this is log scale, it appears that this is 0, but this is not 0, it's just very small starting quantity. So for quite some time we have a lag in turbidity increase, that is a lag in microbial population growth, and then it exponentially rises, undergoes a logistic slowdown, and then stays stationary for quite some time, representing the stationary phase, and then it starts decaying and dying. So this is how population growth looks on a community level. Now this is very important to understand that this is a community level dynamics, not individual microbes dynamics. So an individual microbe will not stay stable and not growing for some time and then have an exponential growth followed by logistic, followed by stationary, followed by death. That's not how microbes grow, but that's how populations behave in lab. So let's look into these each phases more in detail. So first phase is lag phase. Now why do we have this phase, lag phase, where microbes take time to even start their exponential growth? Usually the microbes that we inoculate in our media, they are from a different media, they have been kept in a different environment, they might have different temperature, and they sort of undergo a shock at times, and at times they require an acclimation time, which is the time they require to get acclimated to the new environment. Now remember, microbes are quite finicky, they can be quite sensitive to the environment, so everything makes a lot of difference. So in lag phase, it definitely depends on the history of inoculum, where it is coming from, what kind of media was it in, and what were its growth conditions. Thus, if let's say the inoculum is coming from a very rich media, so that is they have lot of nutrition, lot of carbon sources, lot of sugars to eat and grow, but now I have put them in less rich medium, so here they have less food. So initially they will be shocked, oh my god, where is the food? So they won't kick in an exponential growth. It's also possible that the medium was at different temperature, different pH, and that will further increase the lag phase. Now growth condition is very important. Now let's go back here. So let's say I have two beakers, and in one beaker, which is beaker 1, I have some media here, which is, in other words, very tasty food for microbes, and I have some microbes that are growing here very happily. So this I can refer to as beaker 1. In another beaker, beaker 2, I have some media, but there are no microbes growing in it. So microbes in beaker 1 will follow the model that I just shared with you. So their growth will look like this. Initially, when I first started growth in beaker 1, they had n0 number of microbes. They underwent an exponential growth followed by logistic stabilization. They stayed in stationary phase for quite some time before decaying and dying. So this is their growth rate. Now if I take my a sample from here and inoculate it in beaker 2, the microbes will kick in the similar model here. So let's say I take microbes from when they are in this phase, when they are in their exponential growth phase. So microbes are already, they have a momentum of growth, they are already expressing the proteins required for replication of DNA, required for septum and growth of body, so that they can undergo binary fission. So everything is already ready, they have a nice momentum of growth, and what we will notice is that these microbes, when they are put here, they will have a smaller lag phase. So in this, when I add n0, they will have a very small lag phase, and they will directly start exponential growth and followed by logistic and so on and so forth. But let's say I don't do this, instead I take microbes when we are at the logistic curve. So when this microbial community is undergoing a logistic phase, so they are saying, okay, we are reaching a carrying capacity, so they are slowing down, and now they do not have as many proteins to expressed, so that for replication, so things are quite slow. So now in this case I will notice that they will have a longer lag phase. So they will wait for a while and then they will shoot up in exponential growth. But let's say I take it from stationary phase. So when microbes have already reached their carrying capacity, they are quite stationary, they say, okay, we don't need to grow, we just need to survive and have just enough growth to counterbalance the death rate, and then I put them here, they are not very interested in growing, so they will be like, let's not grow. But after a while they'll realize, oh, our numbers are much lower in beaker 2 and we have a lot of food, so let's grow. So they will take time to do this, and no wonder they'll have a longer lag phase followed by exponential growth. Okay. Now let's say I take microbes when it is in its decay phase. So in decay phase they have consumed a lot of resources, they have produced a lot of, let's say, byproducts that might be, some of them might be poisonous. They have run out of their fuel now, and they're beginning to die. They're past their stationary phase, and this is a very different model. At this stage they will have, and now, so microbes are not in their prime health, the microbial community, and they'll take a very long, and they might hit an exponential curve. Thus, the lag phase depends on the environment that the microbes, the inocula was in, how similar it is. It also depends on how rich it is. Let's say it's very rich and this is not so rich, so the lag phase will be long. It also depends on where in the growth cycle this microbial community was. After lag phase we have exponential phase. Now exponential phase is most desirable for studies. If I want to study something, then I want to capture microbes that are in their exponential growth, because they will have very small lag phase, and they are also very healthy and robust. And this is okay. And after exponential phase we have stationary phase. This is where the growth has arrested, but the metabolism continues. So even though stationary phase shows that there is no net increase in number of total cells, but their catabolism and anabolism, which is basically breaking down food product, breaking down things to create energy, and then storing energy, creating mass. So both of these factors continue. Death may occur, some cells might die, but no overall decay. So there is a difference between death and decay. Death is a negative term in logistic model, decay is when the dn by dt becomes negative. And then they enter death phase, which is a slow exponential death. So eventually they die, but their rate is pretty slow. So what I drew on the board was a straight line, and then actually they undergo an exponential decay, but it's really slow. And this is very important to remember, that these are population level observations and not individual level observations. So dear students, today we studied about population growth and the model that we talked about, a new model, logistic model, and also about different phases when microbial community undergoes first growth, then a stationary phase, and then it dies. And we also studied about different factors that impact how the community will grow and eventually die. And this is all for today. In next lecture we will jump into microbial energetics and the chemistry of microbial communities. Thank you very much.